Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for a speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Andreas Salamos from WuXi Advanced Therapies.
Great, thank you, Leah. So today I'll be talking to you about our Virology Operations group. I'm the Director for Virology Operations at WuXi Advanced Therapies, and I will be going over what cell and gene therapy vector regulatory testing we are able to provide. So in terms of products that we can accommodate at Virology Operations, we provide testing services for a broad range of products, primarily focused upon cell and gene therapy vectors, ranging from the smallest AAV vectors, adenoviral vectors, all the way up to lentiviral vectors. But we also focus on several other biologics and viral products, and that can range from vaccine products or even small monoclonal antibodies, for examples. All of our testing is focused on putting the product through regulatory parameters, and we try to satisfy all regulatory parameters specified in ICH guidelines or the specific market that the product is trying to enter into, ranging from the FDA, the TGA, and all other continents in between. We focus on quantity, stability, and potency, and that can be focused through our infectious titer assays, purity and safety of the product, which encompasses our replication competence assays and our adventitious viral contaminant assays, as well as identity and appearance. And that's primarily focused through our transmission electron microscopy core, which we are proud to offer one of the few GMP-ready TEM facilities globally. In terms of what we are able to provide as support for your assay specifically, or for your product specifically, is to have sustainable testing strategies throughout the life cycle of that product. And what that means is that we're ready to accommodate through our generic offerings in terms of assays immediately upon working with us at WuXi Advanced Therapies from the earliest discovery phases. So when you have R&D products or are currently in proof of principle phases of your product development, we're able to immediately provide testing services focused on quantity, stability, potency, purity, and safety, and identity, just with our off-the-shelf methods. And then upon determining what your specific needs would be, are able to customize and tailor our testing approaches specifically for your product. We also are currently under a restructuring phase and redesigning how virology operations functions. And one of my goals for virology operations is to really grow and have sustainable support through the product lifecycle. And that means that from that early preclinical phase that we can support the product all the way up to market release and commercial release and post-market monitoring. So as the product advances from the drug discovery phase into preclinical studies, we do have GMP method execution ready and our GMP ready facilities. We are focused on providing assay optimization throughout the course of early phase stages of the product and of the assay itself, aiming at high throughput and high capacity accommodation of testing for any lot release programs that would be needed throughout that life cycle. And ultimately, we can establish that assay through protocol qualification, getting us ready for the late phases. So at phase 3, when that product enters and needs a matrix validation, we are able to provide a custom validation strategy, which will prepare you for your eventual FDA filing and review for release into the American market. We also plan on having a sustainable life cycle support for that product by having continual assay support and have SME support for regulatory filings, as well as any additional support that would be needed throughout that product life cycle. In terms of the department specifically, we are under a significant growth phase for the department, and part of my responsibilities as director is to make sure that we are able to sustainably grow throughout 2022 and into the future, so that way we can continue to provide that support for the product lifecycle. One of the things that we're currently doing is undergoing a restructuring and a reorganization, providing a new academic structure to the department. And what that means is it used to be a collective group of individuals working on all of the assays that we provide for testing those regulatory parameters. Instead, what we are currently focused on doing is reorganizing to the point where we have multiple laboratories within a larger department, sort of like an academic structure. So as you can see in the diagram, each of the blue titles are essentially disciplines, or if you want to envision them in an academic setting, almost like labs specifically, where each of the assays within each of those labs would be projects within the laboratory and how they're being run. So in terms of a structure, each of those labs are going to be run by a principal scientist, and then each of the assays themselves are being run by specific scientists. So PhD scientists would be in charge of those almost as if they were projects. We're really focused on the quality of the science. We're trying to make sure that in terms of any handling for that assay or of the product that comes into virology operations, that from the bench top all the way up to management, everyone is considered a subject matter expert for that product and that assay itself. That means that we are able to more appropriately handle any issues that may arise on testing, or if we see anything atypical, ensuring that you have a high quality assay being run and the results are also of a high quality. Part of that is streamlining our training strategy. What we're planning on doing is making sure that everybody can train individually in each one of those labs, in one of the, I call them scientific disciplines for each of those blue categories in the chart. And we're really focused on scientific culture and engagement in the department. So there are constant seminars being held. Something that's very unique to our group is that we're providing everyone within the department to present and learn more and challenge themselves scientifically, so that way they become stronger scientists, both technically in the handling aspect, but then also on their subject matter base. We also have a third or a final group, which you can see in the last bucket, called our value stream group, and that's a group focused on our own growth and performance to make sure that we can sustainably grow and focus on the quality of our testing and services that we provide to clients. And they're focused on making sure that we manage appropriately for our optimization of our assays, as well as optimization of our group, and providing that structure that we need for streamlining our training and providing that scientific culture and engagement. In 2022, part of our growth in what we're doing is we have now brought online one of our first GMP transmission electron microscopy cores. So globally, we're one of the few to offer GMP visualization characterization for thin section microscopy, as well as a negative stain microscopy. And we're also currently planning on pushing through our pipeline of development projects, which is looking at morphology for cell and gene therapy products and or other manufacturing materials. And then we're currently optimizing our AAV protocols. Those will be online the quickest in terms of our pipeline. We're focused on reducing assay footprint for our assays, specifically these AAV protocols that we're focusing on. and improving laboratory processing. And part of that is, again, that sustainable growth. So that way we can positively impact our capacity. So that way we can have more efficient handling in the laboratory and more sustainable throughput for our AAV infectious titer assays and our replication competent AAV testing. Additionally, for 2022, Virology Operations has moved into a new facility. We call this R400 because it's located at Rouse 400. The street name is Rouse. And along with more attractive labs with large windows, we're also impacting our capacity and allowing us to grow, as I've been mentioning. So our capacity is growing for each of our cell and gene therapy assays that we offer currently. So our adenoviral vector testing capacity is going from our current capacity of 2 starts per week to 10. Our infectious titer assays can go up to 21 per week. AAV vector testing can go up to 7, and then our replication competent lentivirus testing, which is currently sitting at 3 starts per week, is able to go up to 14 starts per week. So these new facilities with a lot of brand new equipment is great for us, but then also for all of our clients, as we're able to provide more testing and reduce our backlogs and accommodate a full testing panel for vectoring products. And what that means is that we're able to support any external testing clients similarly to how we would support an internal manufacturing client as well. So in terms of a vector panel of testing, where you may want to submit for an infectious titer assay or a replication competence assay, we can accommodate the full testing panel for your vector product, whether it be an adenoviral vector, an AAV vector, or a lentiviral vector. So an example of that would be if we were to support a lentivector product. The longest assay for that panel of release testing for regulatory requirements is the replication competent lentivirus testing, the RCL culture method. And the reason that it's so long is because there are specific requirements stated in the FDA guidance where there needs to be an amplification phase of 5 passages and an indicator phase, which is a blind passage of the material amplified over those 5 passages onto a new set of cells, followed by a molecular endpoint. Our endpoint is a P24 ELISA, and we do offer an optional PCR targeting VSV glycoprotein. We currently offer at WuXi Advanced Therapies the quickest RCL method. We have the mandatory requirements as stipulated by regulatory requirements and offer the quickest on-test to testing results time frame on the market. It is, however, the longest assay, especially for lentivector testing. So within that RCL testing window, we can also perform the lentiviral infectious titer assay and the in vitro assay for adventitious viral contaminants, which are required for release testing of that lentivector product, all within the same testing window of the RCL method. We are also scaled to be able to accommodate a maximum volume for manufacturing volumes for testing. There is new guidance dictating what supernatant volumes to test for RCL based off of your manufacturing volumes. And traditionally, in the previous guidance, we are scaled to be able to accommodate the maximum of 1 E8 cells from the manufacturing process, which is the maximum compared to 1%, or 5% up to the 300 mils of supernatant harvest. However, the testing requirements have changed, and we are ready to accommodate all of those testing requirement changes as well for the RCL assay. So in addition to being able to accommodate the maximum volumes from the previous guidance, in case there are no dosing strategies known, depending at the phase life cycle of that lenti product, we are able to reduce the RCL supernatant testing volume based off of the new guidance, and we are able to advise on those testing volumes if there is a dosing strategy. The new volumes that are reduced are calculated based off of the dosing strategy in terms of the concentration of the vector, the concentration of cells that are being transduced, or the amount of vector that's being injected, if it's direct injection, and the concentration of that vector at which the transduction is being performed, or transfection, excuse me. The reduced assay footprint by reducing the supernatant volumes on test are significant. It means that instead of testing that 300 mils for the maximum as it used to be, we've seen volumes reduced by about tenfold. And when we see that there are reduced testing volumes for the supernatant testing, we're actually able to accommodate a larger amount of testing, or amount of samples per supernatant material submitted for testing, which means that we're able to accommodate more products more rapidly for a quicker turnaround time from on test to data results being distributed to the client. We have also new initiatives for long-term support as we grow for the assay for RCL. We're planning on implementing a lot of automation. So we collaborate with departments at WuXi Advanced Therapies to make sure that we are on the cutting edge and also sustainably growing, again, with the needs of the client. So we're collaborating with analytical development to automate our capacity bottlenecks, and we're trying to automate our P24 ELISA endpoints, which means that the assay itself is not particularly complex in that it's just cell culture and passaging of cells with your vector product. But the P24 ELISA endpoint is the more complex molecular endpoint. So if we're able to automate that P24 ELISA, we're able to reduce scientific handling time, which will reduce the likelihood of technical errors occurring. And we can also accommodate multiple plates, which means that there's potential for batching additional products, increasing our capacity. Also, because there are clients that may not have patient dosing strategies to reduce the volume of their supernatant testing, we are currently still able to accommodate those maximums of 300 mils, which is a very large assay footprint currently. However, we're currently working to automate and be able to accommodate those large volumes in a less cumbersome fashion, where we can handle that large quantity, but with a reduced footprint, allowing us to streamline that handling and reduce any hiccups in terms of that amplification. And with that, that's all I have, and I'm happy to take any questions. And I appreciate your time and attention.
Okay, great. So what is the benefit of having two endpoints to the RCL method, and why the P24 ELISA specifically?
So I'll actually answer those in reverse. So the P24 ELISA is a bit more reliable and consistent. Oftentimes with the RCL assay, a PBRT endpoint can be offered, but there's a high risk, depending on the vector product or the way that the assay is designed, of receiving a false positive result through a PBRT endpoint. So in terms of consistency, the P24 is a bit cleaner and a bit more consistent. And the benefit of having two endpoints is twofold. One is if the vector product contains VSVG on its envelope, it's an additional data point that can be provided, indicating that there's no recombination event leading to the presence of RCL in that vector product. But additionally, the guidance states, the new guidance states that there can be a transition to a quicker PCR focused RCL method if there is sufficient data to justify not testing using the culture method. So we are providing the VSVG endpoint as an optional endpoint, one, in case the vector product doesn't contain VSVG, but then also to include it as an option if the plan is for that client to eventually transition to just a solely PCR-based method. So that way you have the culture method being run in tandem, and then the PCR data to support those results.
Okay. And what impact did the FDA guidance on RCL testing have on WuXi's method, and how it is being accommodated?
So in terms of the science of the method, it doesn't have a large impact. Our method is still consistent from initiation to the endpoint. The impact that it's had is just reducing the testing volumes. So when we accommodate the 300 mils for testing of the maximum supernatant volume, ultimately the footprint of that assay is very large because we can't accommodate 300 mils in one flask. We have to culture it with a specific ratio on tests, which makes it very large, and you can fill up an incubator quite fast with just the 3 starts that we have as capacity currently, or had currently in 2021. So in terms of how it impacted our assay, we're actually able to accommodate a lot more supernatant testing now with a reduced assay footprint.
Okay, and it looks like we have time for just one more question. If we didn't get to your question, we will be passing those questions on to Andreas, and he can follow up with you directly. So the last question for today's webcast is, how can WuXi's RCL by culture method support a transition to a shorter PCR-based method?
Yeah, so as I mentioned, we do offer the VSVG endpoint, which can add that additional data point of a PCR-based result from the culture amplified material, which supports that additional data point to then build enough of a data set that then you can justify transitioning to a PCR-based method. The only challenges would be based off of the specific vector design, whether or not the envelope contains VSV, and we are able to accommodate design of a custom strategy depending on any alternative vector envelopes that the product may have incorporated.
Okay, thank you, Andreas. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.